Hello and welcome to Science Views from the Valley. I'm your host, Dr. Kelly Kassan. Science Views from the Valley is a monthly program that will explore interesting science topics and how they relate to the San Luis Valley and the Upper Rio Grande region. Hello and good morning. This past Monday, the 21st, marked the start of summer. As summer heats up here in the San Luis Valley, it reminds me of the different living organisms that are able to survive in extreme environments that would kill most other living things. These organisms, called extremophiles, are why I still feel that life on other planets or moons may be possible even in our own solar system. So what makes an environment an extreme environment? An extreme environment is any environment that has a biological factor, temperature, chemistry, salinity, etc., falls below the borderline of the range that allows normal organisms to survive. Today I will talk about why most life cannot exist in these conditions and what extremophiles do in order to survive these extreme environments. Nearly all living organisms, bacteria to mammals, require oxygen in order to survive. The reason is because classic cellular respiration requires oxygen in order to proceed with the reactions that will ultimately produce ATP, adenosine triphosphate, which most living things use as cellular energy. Even bacteria, which lack the ATP-producing mitochondria, can produce ATP. Yet many biologists think that the first living organisms on Earth were anaerobic bacteria that were actually poisoned by oxygen. Anaerobic bacteria still exist today. Though many are facultative anaerobes that can live with or without oxygen, some obligate anaerobes, which are poisoned by oxygen, still exist. These bacteria species are found in deep ocean waters, landfills, deep soil sediments, and even in the intestinal tracts of mammals, including humans. Clostridium botulinum, the bacteria that produces the toxin that causes botulism, Clostridium difficile, an intestinal pathogen that causes diarrhea in humans, and Clostridium tetani, which causes the human disease tetanus, or as it's more commonly known, lockjaw, are examples of these obligate anaerobes. So what did these early Earth bacteria and modern obligate anaerobes do in order to produce ATP instead of oxygen? They use anaerobic respiration, which uses nitrate, sulfur, or some other electron acceptor instead of oxygen during cellular respiration. Some anaerobes may use another anaerobic process, fermentation, instead. So why did aerobes, organisms that require oxygen for their metabolism, become the dominant form of life? One reason is because oxygen is a far more efficient electron acceptor than other elements or molecules, allowing the organism to produce more ATP more quickly. Another reason is that obligate anaerobes lack the enzymes that detoxify the hydrogen peroxide and other free radicals that are produced by reactions involving oxygen. Hydrogen peroxide and hydroxyl radicals damage living cells even in aerobic organisms. But aerobic organisms can produce enzymes like catalase to break down these damaging waste products. When photosynthetic organisms, like the cyanobacteria, appeared on Earth, they produced oxygen as a waste product, which increased the amount of oxygen in the Earth's atmosphere. The higher amount of oxygen in the atmosphere eventually killed many of the early anaerobic species in the Paleoproterozoic event known as the Great Oxidation Event. Anaerobic species today live in areas where oxygen is still scarce. So what kind of life is still anaerobic? Bacteria, like the Clostridium species I mentioned earlier, are one example. Some fungi are also obligate anaerobes, including the fungi found in the rumen of herbivorous animals. These anaerobic fungi play a role in the degradation of cellulose, which allows herbivores to utilize the sugars produced by the breakdown of the cellulose. Yeast used in wine or beer brewing process are another example of obligate anaerobes. Some single-celled eukaryotes are also obligate anaerobes. But as of 2012, no animal species has been found to be truly an obligate anaerobe, though some worm species have adapted to low oxygen environmental conditions. Temperature is another factor in extreme environments, either too high of a temperature or too low. Nearly all living species live in environments that range from freezing to just under 131 degrees Fahrenheit. High temperatures denature proteins, causing them to lose their proper shape and become non-functional. Nearly all organisms have a metabolism based on enzymes, of which the vast majority are proteins. So in high heat, these protein enzymes lose their shape and function, which disrupts the cellular processes of living organisms. Another reason why high heat kills most organisms is because it causes irreversible cellular damage and cell death. But in 1969, biologists discovered the bacterial species Thermus aquaticus. This species was found in the lower geyser basin of Yellowstone National Park and thrived in temperatures of 149 to 158 degrees Fahrenheit. These rod-shaped bacteria created a breakthrough for biologists. Not only was Thermus aquaticus able to live in temperatures long thought to be unable to sustain life, but this species contained a number of thermostable enzymes that we use today in molecular biology. The most famous of these thermostable enzymes is Taq polymerase. The word Taq is taken from the first letter of the genus name, Thermus, and the first and second letters of the species name, Aquaticus. Taq polymerase is a type of DNA polymerase, an enzyme that is used to replicate DNA strands. Taq polymerase remains active even in temperatures that reach 167 to 176 degrees Fahrenheit and does not denature even at temperatures of 194 degrees Fahrenheit, which is very close to the boiling point of water at 212 degrees Fahrenheit. This made Taq polymerase ideal for the process known as polymerase chain reaction, more commonly known as PCR. To replicate DNA in the PCR process, high heat must be used to separate the two DNA strands that are wrapped in a double helix. Taq polymerase can still replicate DNA strands even at these high temperatures. Taq polymerase became so useful that Science magazine, one of the top scientific journals, named it the Molecule of the Year in 1983. Kary Mullis, the biochemist who invented and developed PCR, won the Nobel Prize in Chemistry for his work in 1993. Since then, other thermostable enzymes have been isolated from Thermus aquaticus, which have enabled researchers to further study chemical reactions under conditions where most other enzymes would denature and lose function. Other thermophiles have been found at Yellowstone National Park. The bright colors of the Grand Prismatic Spring are the result of microbial mats, colonies of different species of thermophilic bacteria and archaea. Archaea are single-celled prokaryotic organisms that are related to bacteria. Currently, the record holder for extreme heat living is Methanopyrus candeleri, a species of archaea that can live and reproduce at 251.6 degrees Fahrenheit and can even reproduce in media that is being autoclaved. Thermophilic fungi have also been discovered. Their heat-stable enzymes are currently being studied for possible pharmaceutical use. On the opposite end of the thermometer, some organisms prefer it cold, thriving in temperatures of -4 degrees Fahrenheit or colder. Most living species cannot live in freezing temperatures because our cells are mostly water. Human cells, for example, are approximately 65% water. When the water freezes, the ice crystals will tear into the cell membranes, causing the cells to rupture and die. Extremophiles, living organisms that live in extreme cold conditions, have several physiological adaptations to cold environments that prevent the formation of ice crystals within the cells. One adaptation is the ability to produce polyhedric alcohols and antifreeze proteins in their blood and cells. These compounds, because of their ability to prevent cellular deformities, are being investigated for potential use in the frozen food industry. Unlike the thermophiles, psychrophiles are not only found among bacteria and archaea, but are also seen in fungi, protists, and even insects. Snow fleas, for example, are tiny wingless invertebrate animals that will jump around on the snow surface. They are not actually fleas. In fact, snow fleas are not even closely related to fleas at all, and they are not parasitic like fleas. Snow fleas are springtails, tiny, six-legged arthropods that gets its name from the little spring-like structure under their abdomen, which is how they leap about on the substrate surface. We have snow fleas in the alpine regions of Colorado. You may have seen them during a winter hike when the snow covers the ground. These snow fleas will appear like a group of what seems like dark specks jumping around on the snow surface. Another psychrophile that lives in Colorado is the winter or snow scorpion flies. These insects belong to the group Boreas. These wingless insects, so named due to the upturned abdomen tip that resembles a scorpion's stinger, are so adapted to cold temperatures that the heat from a human hand will kill them. Another type of extremophile is the halophile, organisms that can live in environments that have a much higher salt concentration than our oceans. These environments, like the Great Salt Lake in Utah and the Dead Sea in the Middle East, have salt concentrations much greater than the 35 parts per thousand that we see in ocean water. 35 parts per thousand is approximately the same as 1.4 teaspoons of salt per cup of water. But halophiles prefer a higher salt concentration. Some live in environments that have as much as 209.5 teaspoons of salt per cup of water. This fact is even more amazing considering that high salt concentrations cause cells to dehydrate. Water will leave the cell to dilute the higher salt concentration solution found on the outside of the cell. This water loss will eventually lead to cellular desiccation or cell death. Halophiles are able to survive these high salt environments by using one of two strategies. The first is the use of osmoprotectants, which is the most common strategy used by halophiles. Osmoprotectants are small carbon-based molecules that have a neutral charge and low toxicity at high concentrations. Because they have a very low toxicity to the cell components and intracellular proteins, osmoprotectants are also called compatible solutes. These osmoprotectants accumulate in cells and help to balance the difference between the salinity of the solution on the outside of the cell and the solution inside the cell. This balance prevents the loss of water. These osmoprotectants work so well that some halophiles, like the brine shrimp and tardigrades, can become completely dried out and enter a state of suspended animation called cryptobiosis. Examples of osmoprotectants include amino acids, which are the building blocks of protein molecules, and different types of sugars. The second method is the selective absorption of potassium ions into the cytoplasm. This strategy is much rarer and seen only in a few halophilic bacteria and archaea. The reason for this strategy's rareness is because it requires the different components of the cells and its enzymes to be adapted to the higher concentration of potassium salts, while the use of osmoprotectants do not require these adaptations. Halophiles are so adapted to these high salt environments that they now require salt for growth and will die if they are placed in environments where the salt concentration is considerably lower. Though most halophiles are either bacteria or archaea, halophilic protists, fungi, and even some animals have been discovered. Soy sauce and sauerkraut are fermented salty foods that use halophilic bacteria to facilitate the lactic acid fermentation process. Salt is toxic to plants. Salting fields as a way of destroying your enemy's crops has been used since biblical times. Yet a few plants, known as halophytes, are very salt tolerant. Mangrove trees, for example, excrete excess salt through their leaves, so they can live at the ocean shores. Salt marshes have a number of halophyte species, including cordgrass, sea purslane, and saltworts. Halophytes, because of their ability to remove salt from the surrounding soils, are being used to remediate soils that have a high concentration of salts. Examples of halophytes in Colorado include Ephedra viridis, commonly known as Mormon tea, greasewood, and shadscale. Halophilic animals also exist. The Great Salt Lake in Utah is uninhabitable to nearly all animal life, but brine shrimp and brine flies are common there. Brine shrimp were popularly sold in the 1960s and 70s as the novelty item sea monkeys, which are still sold today. Though higher animals are not halophilic, some have adapted to higher salt concentrations. In 1832, a dry stone dike was constructed on the North Ronaldsay Island, located far north of Scotland. This dike completely encircled the island and separated a herd of short-tailed sheep from the fields within the dike. The sheep changed their diet to seaweed, which contains a high amount of salt. Today, nearly 190 years later, these sheep, now known as North Ronaldsay sheep, have adapted to handle salt concentrations that would kill any other breed of sheep. Another example of extremophile are those that can survive in extreme pH environments, either low pH, acetophiles, or high pH, alkaliophiles. pH is the measurement of hydrogen ions in a solution. A solution that has more hydrogen ions than hydroxyl ions will be acidic. Solutions that have equal concentrations of hydrogen ions and hydroxyl ions are neutral, and those that have fewer hydrogen ions than hydroxyl ions will be alkaline. Most life prefer to live in neutral or weakly acidic or alkali environments that have a pH ranging from 6 to 8. But some environments, like sulfur pools, geysers, or regions that have been polluted by acidic mine drainage from either metal or coal mines, can have a pH of 1 to 5. Soda lakes, like Mono Lake in California and the soda lakes located near Fallon, Nevada, have a high pH, nearly 10 on the pH scale. Alkali Lake in southeastern Oregon has one of the highest environmental pH, a pH of 11, which is about the same pH as ammonia and bleach. A low or high pH will interfere with the normal biochemistry of living organisms, interfering with their cellular function, respiration, reproduction, and other functions. Most acetophiles, however, adapted to a low pH by evolving mechanisms that can pump hydrogen ions out of the cell, keeping the cytoplasm near a neutral pH. A few acetophiles, the bacteria Acetobacter is one, have an acidified cytoplasm, which means the cellular proteins and other intracellular molecules have evolved acid stability. Acetobacter, because of this ability, are frequently used as fermentation starter cultures. Most acetophiles are bacteria or archaea, but a few fungi, like the mold Merkur and the green algae Dunalia, are also acidophilic. Alkalophiles, however, have a different issue. They do not have too many hydrogen ions. Instead, they have too few. Alkalophiles use the strategy of maintaining an acidification of their cell's cytoplasm through either passive or active means. Passive acidification is having cell membranes contain acidic polymers. These acidic polymers help protect the cell membrane by preventing the entry of hydroxyl ions. Alkalophilic bacteria have acidified peptidoglycan, the substance that makes up the bacterial cell wall, which also protects the cell membrane from hydroxyl ions. These passive methods work well in alkali environments that are approximately 2 pH levels above the cell's pH, but cannot maintain this pH in extremely alkaline environments. Alkalophiles in extremely alkaline environments use an active method of acidifying their cells by using sodium-hydrogen antiport proteins to transport hydrogen ions into the cell in exchange of driving sodium ions out. Mutations that lead to a disruption of the sodium-hydrogen ion antiporter results in the bacteria no longer being able to live in alkaline environments. Alkalophiles produce enzymes that have found use in the detergent industry as detergent additives. Since soaps and detergents tend to be alkaline, alkalophilic enzymes are far more efficient than those that are neutral because they can remove protein-based stains like blood, grass, and sweat in alkaline solutions. The ability of living organisms to survive and adapt to extreme environments is rare, but the study of these organisms and the strategies they use to survive these environments have been very useful for understanding how humans may survive harsh conditions during space travel. These examples of extremophiles also give astrobiologists hope that life will be found on other planets and moons. I hope you enjoyed today's topic. My next show will be on July 22nd and will discuss the defensive strategy of aposematism, the use of coloration to warn predators from distasteful or poisonous prey, and the exploitation of that strategy by harmless organisms. If you would like to hear a scientific topic, contact me at spiderwomankrza@gmail.com. If you would like to hear the audio of my earlier shows, all episodes of Science Views from the Valley are archived on the YouTube channel Science Views from the Valley. I would like to thank the people here at KRZA for their support, and thank you for listening.